as you know that we are discussing about the NEET biology, and step by step the photosynthesis chapter we are discussing about for class 11 students. Here we have already discussed all the questions of C4 plants. Now we are going to discuss about all the questions of CAM and C4, CAM and photorespiration. Now here the CAM plants, you know that a CAM is a crassulacean acid metabolism, and C photorespiration, your photorespiration. Okay, now here step by step we are here going to discuss about all the questions of your lineage biology, of your alane, of your Akash material. So you can get all the questions from here of CAM and photorespiration of previous year questions, as well as analytical questions, as well as assessment reasons. So keep patience and take all the type of questions of this particular topic, that is CAM and photorespiration. Know that out of the photosynthetic questions, of the photosynthetic questions, the 30%, more than 30% questions are coming from CAM and photorespiration. 50% questions are coming from your C4 alone, and only to 20 to 30% questions are coming from the C3 part. So let's start the CAM and photorespiration. First question here: CAM plants helps in the disease resistant reproduction, conserving water molecule, and secondary growth. Now your CAM plants are crassulacean acid metabolism, and they are the fleshy leaf plants. We know that the fleshy leaf plant, so fleshy leaf plants actually they absorb much amount of water. So this one is fleshy leaf, that is, the water retention is an important character of this type of plants, that is conserving water molecule. Okay, now here the second question is not related to photorespiration. Here the second question: which organelle is not related to photorespiration? We know that the term CPM, chloroplast, peroxisome, mitochondria, they are sequentially arranged. The phenomenon are sequentially arranged, not only this case. So here lysosome is odd one. This is not related to photorespiration. Okay, now product of photorespiration, you can see that the product is phosphoglycolate, your erythrose 4-phosphate. Not erythrose 4-phosphate nowhere is present in photorespiration. In dihydroxyacetone phosphate, dihydroxyacetone phosphate is also absent here. All of the have not. Here the phosphoglycolate, and the product of the phosphoglycolate is the 2-carbon compound. So why it is termed as 2-carbon compound cell, or C2 cycle. Now photorespiration is also called C2 cycle, as it is first stable product is 2-carbon compound phosphoglycolate. Okay, so this is most important, and these are not. Okay, the substrate of photorespiration is, that is the substrate of photorespiration. We know that the pyruvic is not a substrate. RUBP and oxygen together constitute the 5-carbon compound. RUBP produce 2-carbon compound, a compound glycolate, that is phosphoglycolate. Okay, so this is a substrate main. Now here the CAM plants not so photorespiration because performing Kelvin cycle at night. This is most important. There is a C4 plant is not able to perform the photorespiration, but CAM are also get rid. They also are protected from photorespiration. How? Because their mechanism. Okay, then during the daytime their stomata are closed, and during the night time they open. They absorb carbon dioxide, and they absorb carbon dioxide and they convert it into the malic acid, and that particular malic acid is the main source of carbon dioxide in C3 Kelvin cycle. So during the night time, so Kelvin cycle occur all time. Now this question, this option is very important, because performing Kelvin cycle at night, it is not right, because Kelvin cycle occurs all the time. So only night it is not possible. Using PEP carboxylase only, CAM plants so photorespiration, not so photorespiration during the PEP carboxylase. Not only this, your C3 cycle itself, it has a C3 cycle. So C3 cycle it is also, CAM plants are known as night C4 plants. Night C4 plants. So like that is C4 plants, they also have a Kelvin cycle point. Okay, so that particular Kelvin cycle is photo independent. So during the night they occur actually. During the daytime no carbon dioxide is actually there. So during the night time your organic acid formation occur, and that organic acid is utilized during the daytime, and during the daytime the stomata closed, so oxygen is not coming here. So this is a very important point, that is fixing carbon dioxide into organic acid during the night and releasing them into the carbon dioxide during the daytime. Okay, for Kelvin cycle. So Kelvin cycle here in this case is unique, that is uniquely operated. Normally Kelvin cycle actually is affected by oxygen, but in this case oxygen is absent here, because during the night time they absorb carbon dioxide by the PEP carboxylase and convert it into the organic acids, and during the night time, during the daytime, they use this organic acid and convert, they release the carbon dioxide, and the Kelvin cycle run. So this is very important point. During the daytime the Kelvin cycle is not affected, because during the daytime stomata is closed. So this is very important here, point, that is fixing carbon dioxide in organic acid. So these are the questions of CAM and photorespiration. Now the next questions we are discussing about later, and lots of questions. So let's start. So the next question is, the first reaction of photorespiration is, what type of reaction it is? The RUBP is attached with oxygen molecule, and this is the oxygenation type of reaction, and the enzyme is ribosco, and its oxygenase activity is active here. Now the first reaction of photorespiration is oxygenation, not other than. Now the second question is, CAM plants belong to which family? We know the Crassulaceae family is the family of the CAM plants. Next question, photorespiration is not detectable in which? Where photorespiration is not detected? In C4 and CAM and C4 both. In both case the photorespiration is not detected. Okay, now the photorespiration, how many molecules of glycines are required to release 1 mole of carbon dioxide? This is very important thing, that carbon dioxide is released instead of attached in this case of photorespiration. So here carbon dioxide is released in where? 2 molecules of glycine attached to produce 1 molecule of serine, and during this step 2 molecules of glycine, here the 2 molecules of glycines are required to release 1 molecule of carbon dioxide. So there the answer is 2 molecules of glycines are required to release 1 molecule of carbon dioxide. Now the CAM plants organic acids content, during the organic acid content, that is the malic acid concentration, is increased during the night. So during, it's the answer, that is the CAM plants organic acid contents is increasing during the night. Okay, it increases during the night. So next question, the photorespiration peroxisms helps in, peroxisms CPM, chloroplast peroxisome mitochondria. So peroxisms helps in what type of? Oxygenation of glycolate. So two step of oxygenation occur. First step of oxygenation occur is RUBP oxygenation, and second step is oxidation of glycolate. Mind it, after formation of phosphoglycolate enters into the glycolate. It converts into glycolate, and glycolate enters inside the peroxisome, where it will be converted into glycine through glyoxylate. So after glycine formation, glycine will enter into the mitochondria. So in this case, the oxidation of glycolate. Now photorespiration involves oxidation of what? Photorespiration involves oxidation of RUBP, not PGA, anything, RUBP. Now the photorespiration oxygen consumption reaction occurs where? In photorespiration, oxygen consumption reaction is mainly occurring in stroma of the chloroplast and mitochondria. No, here actually the stroma of the chloroplast and mitochondria. Here these are the most important term here, that is where the photorespiration occur. Oxygen consumption occur where? In photorespiration, oxygen consuming reaction occurs in stroma of the chloroplast and mitochondria. Here you have to remember that so here the Photorespiration, oxygen consuming reaction. During the photorespiration, oxygen is consumed when which is stroma of the chloroplast and peroxisomes. Okay, stroma of the chloroplast and peroxisome in two steps. We know that the RUBP when attached with oxygen and glycolate when attached with oxygen. So glycolate when attached to oxygen in peroxisome, and in chloroplast RUBP is attached to the oxygen. So now next question, which one is cam plant and maize, your pineapple, onion and pea? We know that cam plants are pineapple. You know, the fleshy leaf plants is pineapple, and is the pineapple is cam plants. Maize is actually C4 and these are C3 plants. Source of carbon dioxide for photosynthesis in cam during the day. During the daytime it is malic acid, and during the night stomata open, so easily from environment carbon dioxide is entered, but during the day stomata is closed and malic acid will supply the oxygen. So these are the questions. Able to prevent photorespiration, it is also right. Cam and C4 plants is able to prevent the photorespiration due to PEP carboxylase enzyme and due to its special traits. They are called night C4 plants is also right. They are called, cam plants are called night C4 plants, and the stomata closed during the night. No, this is not right. Stomata are open during the night. So carbon dioxide assimilation and carbon dioxide absorption has actually occurred during the night, and malic acid formation takes place. That is utilized during the daytime. However, this is the wrong about C4 plant. This will be the answer. Okay. Now, which is not true about C2 cycle? Which is not true about C2 cycle? That is the photorespiration. You know, the C2 cycle is photorespiration. It is also called photorespiration. Okay, it is also called photorespiration. Rubisco enzyme is first oxygen acceptor of the cycle. Yes, Rubisco enzyme, in this case, Rubisco enzyme's carboxylase activity is shut down, and oxygenase activity is on. So Rubisco enzyme here is oxygenase enzyme, not carboxylase. Okay, the 2 molecules of glucose is required to release 1 molecule of carbon dioxide. 2 molecules of glycine. 2 molecules of glycine is required to produce 1 molecule of carbon dioxide. Yes, it is right. Also, now oxygenation reaction occurs in chloroplast and mitochondria. Here not. Oxygenation reaction is occurred in chloroplast and peroxisome, not in mitochondria. Not in mitochondria. So this is a wrong statement. So this will be the answer D. So in this case, the answer is D. So if you know each and every point of the photorespiration, you can crack it. Okay. In cam plants, carbon dioxide is stored in the form of what? Cam plants, carbon dioxide is stored in the form of organic acid, which is malic acid. Okay, so answer is malic acid, not oxaloacetic acid or citric acid or both MP. However, the stomata of cam plants, is open during the night. Open during the night and closed during the day. This will be the answer. All are wrong. Now, next question. The C2 cycle, mitochondria. What is the function of mitochondria in C2 cycle? That is in photorespiration. Converts glycine to serine. Yes, converts glycine. 2 molecules of glycine required, 1 molecule of serine. Serine to hydroxypyruvic acid, not hydroxypyruvate to pyruvate, not glycolate to glycolate. This is a function of peroxisome actually. Glycolate to glycolate. This is a peroxisome function. Hydroxypyruvate to pyruvate is a peroxisome function. Serine to hydroxypyruvic acid. Serine to hydroxy. This is also a peroxisome function. So this is how many RUBP required to produce 120 molecules of serine molecule from glycine molecule. You know that 2 molecules of glycine are required to produce 1 molecule of serine. Now we know that after RUBP, 1 molecule of RUBP is produce 1 molecule of glycine. So 2 molecules of to produce 2 molecules of glycine, 2 molecules of RUBP required. So now here from 2 molecules of glycine, 1 molecule of serine is produced. So for 20 molecule of serine, 20 into 2, that is 40 molecules of glycine is required. So from RUBP, 40 molecules of RUBP is required. Okay. Now wrong about photorespiration. What is the wrong about photorespiration? It is a characteristic of cam plants. No, this is a wrong statement. Okay, now it is characteristics of C3 plants. Yes, it is right. It's a characteristics of C4 plant. No, it is wrong. So both A and C, it is right answer. Okay, so in this way you have to answer it. So next question, the C3 and C4 pathway of the CAM plants are separated by, that is, in case of CAM plants are known as night C4 plants. So night C4 plant, why? It's all characteristics like C4 plant. They are protected from photorespiration. They are able to absorb the carbon dioxide directly from the atmosphere with PEP carboxylase and produce the organic acid, oxaloacetate is the first stable product. Now what? In this case, C3 and C4 pathway of the CAM are separated, but in case of CAM, C3 cycle and C4 cycle, these two cycle will run in different places in case of C4 plants. We know that in C4 plants, C3 cycle will run inside the your bundle cell, and C4 cycle is majority is present in mesophyll cell. So in this case, it is separated by not a mesophyll bundle cell. It is a C4 character. Okay, these are not. There's a time. Only the time is a factor. In this case, the carbon dioxide fixation for the production of the your glucose is occurred during the night time. Okay, that is when stomata are open, and here the time that is means C3 and C4 cycle. In this case, C3 cycle is running. C3 cycle is running from where? That is Kelvin cycle during the night time when stomata are open. And in the daytime, C4 cycle continue. Okay, this is the way. And photorespiration occurs in all green photosynthetic parts, or mitochondria only, or roots and stems and leaves only. So all green photosynthetic part photorespiration can be done in case C3 plants. However, now photorespiration is affected by what? Temperature. Photorespiration is affected by temperature, light intensity, oxygen carbon dioxide concentration, and all of the above. So answer is all of the above. That is, photorespiration is highly affected by oxygen carbon dioxide concentration, light intensity, as well as temperature. Okay. Now the organelles, which cell organelles are involved in photorespiration? You know that is the chloroplast, peroxisome, and vacuoles. No, chloroplast, CPM. You have to remember the CPM, that is chloroplast, peroxisome, mitochondria. These are present sequentially from chloroplast, peroxisome, mitochondria. So chloroplast, peroxisome, vacuoles, peroxisome, chloroplast, this one. Okay. Now, if one plant with mutation in rubisco, so that it cannot able to produce absorb the oxygen from the atmosphere, in this plant C4 cycle will run. No, this is a photorespiratory plant. C4 cycle will not run. C2 cycle will be done successfully. No, not able to run successfully because they are not able to absorb the carbon dioxide. No CO2 will be produced. Yes, no CO2 will be produced, and none of the above. No, as photorespiration will not perform, run, so carbon dioxide will also not be produced. So this is a very simple question. First carbon dioxide acceptor of CAM. You have to remember that the first carbon dioxide acceptor of CAM metabolism is acceptor is RUBP. First stable product is oxaloacetate. Okay, so in this way you have to remember.